Good afternoon or morning or evening, depending on where you are, and welcome to the Immunotherapy Treatment Chapter. This is brought to you by the Myeloma Crowd Community Program by HealthTree, and my name is Audrey Burton-Bethke, and I am the Myeloma Crowd Community Manager. We'll be sharing some slides with you today in order to do a couple of intro announcements. Today's session will be recorded and will be sent out to all registrants within 48 hours after the event has concluded. We're grateful to have so many of you joining us and are excited, as you are, to join with Dr. Hultcrantz, who we will introduce formally in just a moment. As always, I would like to thank our sponsors, without whom this would not be possible: Bristol Myers Squibb, Takeda Oncology, Karyopharm Therapeutics, Adaptive Biotechnologies, Oncopeptides, Janssen Oncology, AbbVie, Sanofi, Amgen Oncology, and Genentech. Our event agenda today is as follows. My introductions will last just a few more moments. I will then turn the time over to Myeloma Crowd founder, Jenny Ahlstrom, to speak to us about why we chose this topic for this chapter today. She will also introduce Dr. Hultcrantz. Dr. Hultcrantz will then present her bispecific antibody review for about an hour. And after Dr. Hultcrantz's presentation, we will then open up the session to questions and answers. This meeting is scheduled to be 90 minutes long and will end at 2:30 p.m. Eastern. So Jenny, the time is now yours. Oh, you're muted, I think, Jenny.
I am muted.
There we go.
We appreciate you attending, and we know a lot of you will be watching this in addition later. Dr. Hultcrantz, thank you so much for joining us. We're so appreciative of you doing this for us. Bispecific antibodies are a huge topic. We've all seen, I think, CAR T cells kind of evolve. We have one approved now. Another one may be approved this year. And I wrote an article last ASH on the race for bispecifics, because that's what it seems like to me. There are many, many companies in the space, all doing different targets. You'll hear a lot about that today. But we thought this was a very important topic for this chapter, because learning more about it, when to use it, who to use it for, is just so important for us as patients. So I applaud you for watching this and for making sure that you're up to date on what's happening in the myeloma space. Dr. Monoclonal Hultcrantz is a board certified hematologist. She practices at Memorial Sloan Kettering Cancer Center. She specializes in multiple myeloma and precursor conditions like MGUS and smoldering myeloma. She's focused on identifying optimal treatment strategies for people with multiple myeloma, because not all myeloma is the same. And Memorial Sloan Kettering is one of the largest facilities that are running clinical trials. So I would encourage you, as you're looking at clinical trials, to look at trials, especially if you're on the East Coast, at the center, because they have a lot going on. She's working on measures to continue to improve quality of life while receiving treatment for multiple myeloma. And she has a particular interest in studying the markers of progression from precursor conditions to active myeloma. So Dr. Hultcrantz, thank you so much.
Thank you for that kind introduction, and thank you all for coming to listening to this as well. Okay, I'm going to share my slides.
I think I'll let you present, and then I'll jump back on for to help moderate the Q&A.
Yes.
Okay.
There. So thank you so much again, and that was an excellent introduction, and I think also particularly the idea of how interesting these drugs actually are. So I'm going to start by talking a little bit about the history, or kind of the development of multiple myeloma therapies over time. So what we call the novel therapies, they started somewhere around 2003. The first one was approved, and then as the years have gone on, especially to 2015 and after, we've had a number of different types of therapies that have been approved. Of course, it started with the proteasome inhibitors. We have the IMiDs, Revlimid, Pomalyst. the monoclonal antibodies, daratumumab, elotuzumab, and then lately also the bispecific antibodies, antibody drug conjugates, and as we heard, the first CAR T cell was recently approved. So for the immunotherapies, there are a number of different mechanisms that we have. So a number of these we're going to cover a little bit today. We have the CAR T cells, we have the monoclonal antibodies, we have the bispecific antibodies, and the antibody drug conjugates. Checkpoint inhibitors and vaccines are not there yet and have not shown the same type of efficacy. So we're going to focus on these four groups, and particularly the bispecific antibodies. So quick word about the monoclonal antibodies. These drugs are in what we call a naked antibody, and then they recruit different parts of the immune system or have their own also effect to, in different ways, kill the myeloma cells. Recently, we also have, which was approved last year, belantamab mafodotin. This is an antibody drug conjugate where you have a monoclonal antibody, which is then linked to a chemotherapy agent. So what happens is that the chemotherapy agent gets delivered to the myeloma cell, to this target BCMA, and then it gets taken into the myeloma cell where it releases these chemotherapies. So it really is a very, very targeted delivery of the chemotherapy to the myeloma cell. Side effects with these include ocular toxicity, but they have they're a very interesting group of drugs and a good effect. So CAR T cells, just a quick word about them as well. These are human T cells that have been then recovered from the patient. They've been genetically modified, expanded in the laboratory, and then given back to the patient. And what happens is that they're given this extra receptor. So the T cell, which is part of the body's own immune system, and is supposed to really recognize the myeloma cells and kill them, but the myeloma cells have different ways of evading this normally. So here you really give the T cell an extra receptor to really find and kill the myeloma cells in a very specific way. So as I said, this is a process where you collect the cells and then you reprogram them, you have to grow them, and then it's given back to the patient after what we call lymphodepletion. So you give chemotherapy, and also you have to be admitted usually for days to a couple of weeks. So this is a process. It is a very effective treatment. It's a one-time treatment, and they're durable responses. So coming to the CAR T cell targets, the main target is the BCMA. And as we heard, the first one, ide-cel or Abecma, has recently been approved. Cilta-cel likely will be approved sometime soon. We have other targets, DPRC5D, SLAMF7, and then a number of more also on the way. These are all use the patient's own T cells, but there are also allogeneic CAR T cells where you have healthy and someone's donor cells. So these are what we call off the shelf. So you don't have to go through this process of 6 to 8 weeks to wait and have them reprogrammed and grow in the lab. So these CAR T cells are more ready to use. The side effects of CAR T cells is mainly something called cytokine release syndrome. It's called fever. And what happens is that these cells, they set out a cytokine storm or a cytokine reaction in which the body reacts to these new cells that are in the body. So when we grade this from grade 1 to 2, which is easier, grade 3 to 4 is more significant. Grade 1 usually can be managed pretty easily. Grade 2, we give steroids, we give IL-6 blocking agents, tocilizumab is one of them. Grade 3 or 4, it's more severe reaction and maybe also ICU admission for those. There's another type of reaction also that's called ICANS, where it can affect the nerve system. Usually this goes back, but this happens in 10, sometimes up to 20% of patients. So very good treatment, very good effects, but there are some side effects, and it is a somewhat tough treatment. The bispecific antibodies, they go through a fairly similar mechanism, where instead of having taking out the body's own T cells and reprogram them, you give an extra antibody to bring the plasma cells, or the myeloma cells, in close proximity to the T cells, so that it helps the body's own T cells to recognize the plasma cells and then to kill them in an effective way. So here you see the plasma cell, here is the body's own T cells, and then you have this antibody which brings them very close to each other. There are really two types of bispecific antibodies. And here the naming of these kind of goes into each other a little bit, and it can be somewhat confusing. So the one type is called BiTE, and this is a trademark name by one of the companies. So instead of having the whole antibody, you take the outermost part of the antibody here, and then you link it so to the one that recognizes the plasma cells. So you have one part that recognizes the T cell and one part that recognizes the plasma cells. And here, as I said, you take just the outer part. So this is a smaller molecule, and it brings them in very close proximity. The other type, it's called a duobody, where you, same principle, that you have the CD3, which target the T cells. So you have one arm that here, and then you have the other arm coming from an antibody that recognizes the plasma cell. So you have a bispecific, meaning you have two sites here, one towards the T cell and one towards the myeloma cell. Still brings them in very close proximity, so the T cell can effectively kill the myeloma cell. So I'm going to go through a number of the clinical trials now. So just as an overview, I wanted to show quickly how these trial processes are, because these can be somewhat confusing. So the preclinical data, that's what happens in the lab before any of the clinical studies are done. Phase 1, these trials are really focused on safety and then to find the effective dosing of the drug. So usually they are a dose escalation, where you start at a low dose, see that it's safe, and then you come up to the next dose, see that that is safe, and then you treat usually up to 3 patients per dose level. And then when you see that it's safe, it's tolerable, it's not toxic, then you go up to the next dose level. And after a number of these dose escalation stages, then you find the goal is really to find what we call the recommended phase 2 dosing, which is not too toxic, but still very effective. So there is you want it to be as effective as possible, but not toxic. When you have that dose is found, then you go through to the phase 2, where usually all patients are treated at the same dose level. So there are more patients treated at the same dose level. Usually everyone gets the same treatment. And then if you see that the drug works effectively here, usually go to a phase 3, where you can compare it to a standard of care treatment. And then somewhere, the standard step is to go after the phase 3 to go to the FDA review. In oncology, and oftentimes in myeloma, you can go directly from phase 2 to the FDA if you have really good data and they show good results. After the FDA approval, you need to go through a phase 4 to capture data on a larger patient sets, and also kind of real world data, as it's called. The trial endpoints first, as I said, is safety, and then we have a number of ways to grading the response. Usually the overall response rates, that is partial response or better. So it includes all of these. So that means the partial response is you have a reduction of the myeloma markers. So reduction of M-spike light chains more than 50% from baseline. VGPR is you have a reduction over 90%. Complete response is that the labs look normal. And then you have the minimal residual disease, where the disease is not detectable in the bone marrow. So these are kind of the levels of the response that we look at. So for the bispecific antibody trials, so these, all of them are usually given either IV or subcutaneous. Some of them are given every week, some are given every 3 weeks. As I said, a lot of them are still in the dose escalation phases. And also then the way they're given now in the trials is that the first doses are given inpatient. So the patient is admitted, we run a number of labs, and that everything is okay, and you give the dose inpatient, and then you monitor for 48 to 72 hours, because we know that the side effects, which are similar to the CAR T cells, with primarily the CRS, the cytokine release syndrome, but they're a bit lighter, so they're not as pronounced as the CAR T cells. But that is what we monitor for, and that typically happens within the first couple of days after the first doses. So depending on which trial it is, after around 3 doses inpatient, usually after that, there's no more CRS. So those doses can, all the rest of the treatment can be given outpatient. The targets on the myeloma cells, so these are proteins that are on the myeloma cells, mainly is the BCMA, the B cell maturation antigen. So there are a number of bispecific antibodies, and I'm going to go through a few of these, not all of them. The ones that are more interesting or have come further. So BCMA, mainly teclistamab, elranatamab, and then CC93269. The next target, which also is very interesting, is GPRC5D. It's called, that antibody is called talquetamab, and we also have CAR T cells towards that antibody. There is another interesting one that we also have open at MSK, called FCRH5. So what you really want with these targets is that you want them to be expressed on the myeloma cells at a high level, but you don't want these targets to, you want a target that's not expressed on other cells really. So you want something that is specific to the myeloma cells, so you can have the effect really specifically to the myeloma cells. And that is true for all of these targets, BCMA, GPRC5D, and FCRH5. The patient population, or the participants in these trials, are what are usually called heavily pretreated. So they've seen a median of 5 to 6 prior lines, and around 80% of patients in these trials are what we call triple refractory. So they have been exposed to proteasome inhibitors, Velcade, Kyprolis, or Ninlaro. They have seen IMiDs, Revlimid, pomalidomide, or Pomalyst, and then CD38 antibodies, so daratumumab, Darzalex, or isatuximab, Sarclisa. So for the first one, this is called teclistamab. It's targeting BCMA. It's given IV or subcutaneous. So the first part of the phase 1 was given IV, second part subcutaneous, because that's usually easier. Now this is, so I'm going to show you shortly the results from the phase 1, but these are now in phase 2 trials, and also we are also opening at MSK and called a platform trial, where this drug is combined with several different other drugs. It can be combined with IMiDs, it can be combined with proteasome inhibitors or other treatments for multiple myeloma. In total, there's 6 trials open now with teclistamab. So these are the overall response rates, and this was updated at ASCO now in May, June. So there were 73% participants. All of these were given subcutaneous dosing. The overall response rate was 65. Very good partial response or better was 58. So these are really high numbers for a single agent. So if you compare these numbers to single agent, say with Darzalex or with Blenrep belantamab, the single agent activity in this patient population is usually around 30%. So this group really has a very high single agent activity. The CRS rates, this cytokine release syndrome, was seen in 60 to 70%, which is fairly high, but all of them were grade 1 or 2. So yes, you had to be treated for it, but it's manageable, and all of them they're reversible. The median duration of response in most of these trials have not been reached. So that means that the response is the patients still continue to respond, but bear in mind that most of these have been open in a year or less. They're not very long follow-up times that we have. Next one is called elranatamab, also targeting BCMA. This is weekly dosing. This trial is for 30 patients. This is still the phase 1, so this is the dose escalation. So at doses of 215 micrograms per kilograms or more, the overall response rate was 70. And in this group, there's also 30% complete responses, and 10% of patients were MRD negative. CRS rates 73%, but also here no grade 3 or 4. So here is it's a representation of, this is all, each line is one patient. So here you see the 215 dose, 360, 600, and 1000 microgram per kilogram dose. So here you see these patients have very good response. The responses here, so VGPRs, complete responses, and these little red arrows mean that they're continuing on trial and are continuing to respond as well. So as I said, mostly less than a year, but promising going forward in terms of good responses and durable responses. So as I said, for those that did respond, we see very, very good responses going forward, and this is here up to a year. This is now the recommended phase 2 dose has been decided on, and this is moving forward now in a phase 2 trial. This drug doesn't have a name yet. It's in the stage where it only has this code, CC93269. This is a little bit different. It's also targeting BCMA, but it's what they call a 2-to-1 T-cell engager. So it's an asymmetric antibody where it has two sites where it binds BCMA. So both this site on one side here, and then this outer part here, these are two places where it binds BCMA. And this part here, this is the receptor for CD3 connecting to the T-cell. So this actually showed very, very promising results. In the therapeutic dose, it was up to 90% single agent activity. And of those 13, there were very few patients still, but of the 13 that responded, 92 were MRD negative. So this is a very interesting drug that we're looking forward to seeing more updated results from. CRS rates were around 77%, with here you start to see also the grade 3 patients. So there's one patient that had a grade 3 CRS. So moving from the BCMA targeted drugs, this is other drugs that have also different targets. Talquetamab, this targets GPRC5D. It's a weekly subcutaneous dosing. It's an ongoing phase 1 dose escalation. The recommended phase 2 is now found. So it's more moving forward with 30 patients also at this effective dose for the phase 2. Overall response rate 70%. CRS rates was 67. And then also there was here 1% that had a grade 2. So most of the CRS still manageable with grade 1 or 2. Cevostamab is it's another target, FcRH5, also highly expressed on multiple myeloma cells, not so much expressed on other cells. This is given every 3 weeks as an IV. It's an ongoing phase 1 dose escalation still also. The overall response rates here were 53% at this dose, which seemed to be where we start to see the effect. Going up to a higher dose, it was 61%. So also very good responses from this drug. So here is an overview of where we see more kind of a broader number of, a broader number of these drugs, teclistamab, elranatamab, as I said, the CC93269, talquetamab, and cevostamab. Here you see the target. Most of them still target BCMA. Then we have GPRC5D, FcRH5 regimen, some IV, some subcutaneous. Most of them are weekly, some of them are every 3 weeks. As these are still phase 1, and most of them, there are not very many patients that have been included in the trials yet, but we still see very good responses. So most of them are over 60%, up to 80% and 90% here. As I said, the duration of the response, most of them are not reached, but bear in mind, most of them, the median follow-up is less than a year. The CRS rates around up to 60, but sometimes 70 and 80%. Very few grade 3 CRS though. Neurotoxicity, not so much less than for the CAR T cells. Infections seem to be somewhat of the side effects that we see. And also for those that are given subcutaneously, there are some injection site reactions. So end of the summary of the effect here, we have these treatments are highly effective. Most of them are over 60%. When you get up to some of the therapeutic doses, we see almost up to 100% overall response rates. Main side effects, as I said, the CRS. There are very few grade 3 to 4 reactions. Neurotoxicity, most drugs, we don't see that, but there are a few where we see 1 to 3%. We do see some anemia, neutropenia, thrombocytopenia, 30%, and also leading to some infections. And as I said, injection site reactions up to 25%. So the conclusions here is that these are highly effective treatments. We have many treatment targets. They're manageable side effects, but still they're pretty potent drugs. So we usually recommend, at least for the first doses or for the trials, the patient needs to be admitted. It's a bit too early to say if one of these is better than the other. They all seem to have very good effects. And now that we're getting to the end of the phase 2, starting phase 1 trials, moving to the phase 2 trials, and also moving into combination therapies. So going forward, this, as I said, is really one of the future of myeloma therapies. I actually Googled BiSpecific antibodies for multiple myeloma, and this is one of the images that came up. And I fully agree with this. I think this is a very, very promising mechanism, and it's fantastic that we have so many treatment options. And as we heard, this is really a race to kind of get out there first. I think some of them are getting too close to going to the FDA, and I hope we will have the first one approved within the next year. Thank you. That was not really an hour, but I have lots more to say, and happy to discuss. I see there are already a number of questions. So
Yes, we have a lot of questions and I have a lot of questions. So if you want to, yes, we'll be talking a lot about this. So first, let's ask some broad questions before we jump into the questions that are in the chat. Why is this space so crowded? Like, what is it about the production of bispecific antibodies that makes it so, it seems like it's kind of easier or something to manufacture, but maybe that's not the case. And maybe it's just a great tool, you know?
Yeah, it's definitely easier if you compare it to say CAR T cells. It's definitely easier to manufacture. I think the antibodies. I think what really is the challenge is really finding a good target. I think the actual this is not what I do day to day in kind of manufacturing, but the actual the making of the antibodies, I don't think that is the kind of the limiting step here. I really think it's finding a good target, and then yeah. So I think that's why there's so many variations, and there's so many drugs and companies that want to come into this space.
Okay. Well, okay, I have a lot of more specific questions. Well, maybe we just ask another broad question. So as patients, because these are still in phase 1, maybe some early phase 2 clinical trials. So it shocks me that you say we might see an approval within a year. That's exciting to me as a patient, because what that means is the FDA is not requiring to get them all the way through a multi-hundred patient phase 3 study before approval. Yeah. Do you want to talk a little bit about what you've seen? Because I saw that with daratumumab, and we saw that with a few other, even things like melflufen or things like that were approved after phase 2. Do you want to explain to just our listeners, like how that process usually works? And then I'm really impressed with the FDA. And I think the myeloma group is really trying to get things out into the market as soon as possible. But I was surprised to hear you say that.
Yeah, I think I'm really impressed by the FDA myeloma group as well. I've interacted with them. I know some of them. They're really good data-driven people. Very reasonable. They don't cut corners at all. They're really scientifically driven. But they do also look really at the data. So if you have a phase 2 trial result that is very interesting, and that shows that there's no kind of real safety issue, and you have something with a very good effect. It actually, in oncology and multiple myeloma, it oftentimes actually can be approved after the phase 2. So you said it happened with daratumumab, it happened with carfilzomib, it happened with belantamab mafodotin. So because also they know that there is a need, even though we have all these fantastic new drugs, eventually many patients come to that point where there are no more options, or there are limited options, so we still do need more treatments. So that's why we oftentimes see that actually in multiple myeloma. And I would not be, you can say a lot about the drug from the phase 2, then this is my opinion.
Right, you've already been through the safety.
Exactly.
And now you're doing efficacy, and you have some efficacy data, right?
Yeah, so a lot of the efficacy data you get that we use and that we kind of make the decisions from are from the phase 2 trials rather than the phase 3. Because the phase 3 is also, it's very hard to do, because if you have something effective, you have to compare it to standard of care, and the field is moving so much, so fast forward. So the standard of care can be somewhat outdated at some point. So I think that I think it's very reasonable, and I think it's very good of the FDA to actually look at the phase 2 data.
Okay, absolutely. My last question before we jump into these, because we're getting a lot more questions, so we want to make sure they're all addressed. We wrote an article, or Bonnie Falbo, she's a wonderful myeloma coach and very knowledgeable. So I asked her to pull together an article about comparing and contrasting CAR T therapy versus bispecific antibody therapy. So because these are all in still trials, and to receive this therapy, you need to join and participate in a trial. when you have patients come to you and say, I want to join an immunotherapy trial, which one should I consider when you look at CAR T versus bispecific? What are the main pros and cons or factors that you look at in that discussion with your patients?
I think both are really, really good. I think the CAR T it's going to be more of a process. So oftentimes you need to be a little bit more fit to join a CAR T trial, and fit in terms of both age and biological or age or fitness or whatever you have to, because it is a bit of a process. There are more side effects. You have to go through the CAR T cell collection. Oftentimes you need some bridging therapy because of these 6 to 8 weeks. Then you have to be admitted. You have to go through the chemotherapy. You have to have the potential side effects with the CRS. So it is a bit of a tougher treatment, the CAR T. I think that it's very much a discussion with the patient what to join. If they are able to and fit enough and want to join the CAR T, I think that's an excellent trial. And a lot of patients are actually wanting to go through, take that kind of heavy or more of a I wouldn't say
one and done.
Exactly, it's a one time and then you can be monitored off.
Right.
So it's also really what you prefer. But there are some kind of there are some cutoffs in terms of what kind of physical stage you're in and your
like your lung function and your heart function, all that.
Exactly, that all have to be kind of in an optimal way. So the bispecifics are going to be able to, we're going to be able to use them on a bit broader, because the side effects are a little bit easier. There may be a bit more side effects than say daratumumab or isatuximab, but still very manageable. So you can still use it in a very broader patient group, and there also are off the shelf, as they're called. So we don't have to go through that collection, the manufacturing process. You can just kind of take them and use them now within a clinical trial, but eventually also when they're approved, it's going to be pretty easy to do.
Are there any autologous or your own cell bispecific antibodies?
Not that
They're all off the shelf.
Yeah,
okay. Yeah. Okay.
And I don't think that really, I'm not sure it matters for an antibody really, but that's I'll have to. I think the off-the-shelf antibodies are that's what all the antibodies are. I actually haven't heard of any of the.
Yeah. I hadn't heard it, but it just kind of made me think about that when you're
good question.
Comparing and contrasting. Yeah. Well, there are so many that it's not really needed, right? Okay, there's a question here that I had too about sequencing immunotherapies. So what I'm hearing from all the investigators is, oh my word, we have these amazing new therapies. We're doing the clinical trials on highly relapsed patients. We're trying to move them up as fast as we can to either high risk newly diagnosed or like 1 or 2 prior lines of therapy instead of 3, 4, 5 prior lines of therapy and things like that. So we're going to be using more of these therapies in your treatment lines. How do you go about the process sequencing these immunotherapies one after another? Like I have a friend who did a CAR T and that didn't really work very well, is currently in a bispecific antibody trial, but is potentially maybe even relapsing on that trial. So with a different target than the CAR T. So how do you look at that as an investigator about, I don't know, like, we've had these therapies, like carfilzomib and you do the triplet combination, the Darapom-Dex or RVD or something like that, that are more standard therapies that you've been using in the clinic for a really long time. But what do you do? How do you think about this? In the immunotherapy space? It's very complicated.
Yeah, it is very complicated, and I don't think there's a straight answer to it either. I think if there isn't, the CAR T is still a little bit kind of, there's several different answers to that. I think that that there's no real way that we can say that starting with BCMA targeted or GPRC5D targeted is better than the other. You can certainly have a really good response to if you relapse from BCMA targeted therapy to one of the other drugs. There's also a lot of evidence because BCMA is so widely expressed on myeloma cells, so you can still respond even if you get a CAR T towards BCMA or belantamab mafodotin, which is also targeting BCMA. There's still a good chance that you're going to respond to one of the other BCMA targeted drugs.
Yeah, because it's not level of BCMA; it's something else that's going on.
Well, there's a number of different things, and also there is so in terms of the BCMA particularly, there is some evidence that there is one of the ways of kind of the mechanism of resistance is that the cell gets rid of BCMA from the surface. And if the therapy then disappears, then the cell recovers.
There's nothing to go after.
Sorry?
There's nothing to go after at that point, right?
Exactly. There's nothing to go after, and then there's there's a resistance mechanism, and the drug doesn't work as well. But if you then stop the drug, a lot of times we see that BCMA is actually coming back. So that's why you can respond again. And there are also other ways, we're looking into that as well, where you can have ways of enhancing the BCMA on the myeloma cell to get to the cells to express even more BCMA, or not cut the BCMA from the cell surface for the BCMA targeted drugs to work even better. So there are some ways of getting around that as well. But then I think in terms of, so there's no straight answer in terms of how to sequence these. In terms of a practical more approach right now, when most of the things are still in clinical trials, I think if there is a possibility and the person in question can do a CAR T cell, I think that's smart to do. And then, because that's a little bit, with the availability right now, it's a little bit harder to come by. So I think that's smart to do if it's available, because we know that the bispecifics, we have so many trials, and there's so many centers that have many trials. And as I said, like they're gonna start being approved fairly soon. And then it's because even though Abecma, the ide-cel, the CAR T cell is approved, it's still a process. We still only have kind of a certain number of patients that we can do at a time with that. So I think
we're trying to ramp up manufacturing and all that, the whole process.
Exactly.
Like you were saying, and only certain centers can do it. It's not like you can go to your local cancer center and maybe they'll be able to do it for you, maybe not.
Exactly. So I think in terms of if there's a chance that you can that is available, I think that's smart to do. And then the bispecific, I think, are really good. But if it's kind of being a little bit more pragmatic, that's easier to get to than the CAR T. So
yeah. So maybe,
but moving forward, kind of in the bigger picture, I think these are really going to move up as well. So the way, as you know, with both with carfilzomib and daratumumab, they started in the same space, and then we see that they're really effective, and then we move them up to the front line. So in a number of years, these are going to be specifically the bispecifics are going to move up pretty quickly, I think, in terms of treatment where we use it. And going to be combined with KRD, VRD, whichever combination you have eventually as well. And then the CAR T, I think, are really going to start challenging the transplant in that space.
That's the feeling I get. I keep hearing that from myeloma investigators, because like, I guess you could always go to transplant later and you could try maybe a CAR T or something up front, and then use transplant as your second line of therapy or something.
Yeah.
Interesting. Okay, Graham asking a question about BCMA expression. So I think you mentioned this, that BCMA is a target on multiple myeloma cells, most multiple myeloma cells. That's why it's such an attractive target. It is found on normal B cells also. So is there any downside? Is there any damage that it does to the immune system in general by targeting BCMA that you've seen?
not more specific than most of the other therapies that we have. So they do give some immune suppression. We do see some cytopenias with anemia, neutropenia, thrombocytopenia, and there are some infections. But that we also see, we see that with daratumumab, we see that with VRD, KRD, we see that with most therapies. So I wouldn't say that there are anything specific that we see. There's some treatments, say for lymphoma, where you kind of get a more of a B cell depletion that can be going for a very long time. There's nothing really that we see in that sense.
Yeah.
No, nothing more really very much specific and more than general immunosuppression and sensitivity to infections.
Okay. Okay, Asbeli has a good question. This is a great question because you reviewed like CAR T versus bispecific in terms of strategy. So if you're a myeloma patient, and I experienced this because I need to make a treatment decision, I've chosen one and I'm going to do that. But this was what went on in my brain when I was going through it. Do I do a standard triple combination therapy and wait for some of these things to mature? Do I do an immunotherapy and then know that I can go back to those triple combinations later? Like when you look at, do I do Darapom-Dex? Do I do KRD? Do I do those things? Or do I try to jump into a trial and use it earlier? It kind of goes back to the question about like the status of your immune system. Like at what point is it too exhausted to do this? So, and then we can kind of maybe talk about T cell exhaustion and that concept.
So it's a really good.
It's a hard question.
I don't have a straight answer to that one either. I think that is there's no right or wrong. I think the trials, I think all are most trials are really interesting. So if there is a chance to join a trial and your doctor recommends that, then I would probably go for that, because I think that there's some trials, and that's kind of how I have the discussion with my patients of like this, if it's something that you can have access to that you would not outside of the trial, then that's a good thing to do, I think. Also because the trials, the standard treatments, you can always fall back to, they're always going to be there. The trial treatments, if there are 30 patients, a lot of these are competitive enrollment. The slot is not going to be there if you change your mind in 6 months or so. So I think that is one thing. That said, no one should be pressured into joining a clinical trial. It's very much, it should be completely voluntary and so on. But I think most clinical trials are interesting and worth pursuing if that's an option. In terms of the T cells, so if you look at the patients the trials that I presented now, most of those had gone through 5 to 6 treatments on average. So by that point, your T cell response and your T cells are already pretty tired, and you can see still that they're very good responses. So moving that up forward, we're probably going to see even better responses. Both because the myeloma cells are kind of more naive, they haven't seen as much therapy, because know that as kind of the disease goes on, the ones that the myeloma cells that are still there are the more kind of the ones that aren't taken out by the other therapies. So they are kind of the more aggressive or stubborn ones that are still there. And these seem to be, even in that setting, have this single agent effect of 60 to 70%, which I'm really impressed
by. It's really high response rates, right?
Yeah.
For a single agent.
So using that earlier, I think we're going to see even better responses. Because of the myeloma cells being not as they haven't seen as much treatment, they're not refractory, and also because of the T cells are going to be not as exhausted.
Okay, Mariana is asking a good question. It's specific to translocation in 1114, but I think this question applies to all the genetic features in myeloma. Have you seen, I mean, the phase 1 studies are so little numbers of patients that you probably don't know the answer to this yet until you get the phase 2 or more patients on the trials. But are the bispecifics showing more efficacy in certain genetic features of patients like 1114 or others? Or is
yeah,
it like
we don't know that yet. Yeah, we don't know that yet. And we do know that they work in both standard and intermediate and high risk, but there is no data that I've seen where it's better or worse or so for any of the specific translocations or genetic changes. It also, we usually don't look at the surface antibody kind of expression before we start. So we just kind of assume that they're there and that they're going to work. But going forward, we'll probably look even more towards the surface expression and then also subtyping. And that's a good thing that we have so many, so we can kind of see which one works better. But we don't have that data yet.
Okay. Vicky's asking a question about side effects. So I know you said the side effect profile is lighter in general than the CAR-T. I've had friends say, oh, I thought it was going to be so much lighter because that's what everybody talks about. I've had, like, I have to stay in bed for a few days after this trial that I'm on, and Vicky's asking about talquetamab specifically and fingers and nails. And is that common? Is it reversible? Is it painful? This is this is steep learning curve, I think, for everybody, right? Just getting the names right, first of all.
Yeah, that's
on all of them. The certain side effect profiles, can you speak to side effects and just what you've seen in the trials you've been running?
I think the in terms of fingers and nails, I think that I have seen that with patients on various treatments throughout. I don't think, to my knowledge, and we have some of these open, we don't have all of these open at MSK. So I can't speak to my personal experience with all of them, but that's not something that I've seen a lot of. But yes, it is a very potent treatment. As I said, for the first around 3 doses, you have to stay 3-4 nights in the hospital. We're monitoring very closely, and then yes, it's a potent therapy. It is exhausting. It's not, it's more side effects than, you say, the monoclonal antibodies. So yeah, but it's less than CAR-T, but it's not just a walk in the park.
It's not nothing. Right. Oh yeah, and they will let you know if you're participating in a trial, kind of what you can anticipate also. Okay, so Natalie asked, can you target specific cytogenetics with these? And the answer to that is really no, right? Because you're targeting this expression.
Yeah, these proteins that are targeted, they're on the surface
of the myelomas.
And the genetics are they're in the nucleus. So that's kind of inside of the cell. So in the myeloma cells, they tend to look similar on the surface, regardless of what the genetics are inside of the cells either way. That said, as we go forward, obviously, we're going to see much more of if there are any specific subgroups there where we see better or worse effects.
Okay, Belinda and Hatham are asking a similar question. If you try a bispecific therapy and you start relapsing, can you do another trial with a different target on another bispecific antibody? Yes.
So you can even we have a number of patients that have done, say, belantamab, they have gone through CAR-T, they've done a bispecific, all with BCMA, but then you also have the GPRC5D and the FcRH5. So you can certainly do several ones with the same target and also then move to other ones. So yeah, we have several patients that have gone through clinical trials with bispecific different targets for sure.
Well, the hope is wonderful. So being able to see something else that you could try is fantastic. So Heather's asking a question that's very interesting. Are these are the bispecific antibodies applicable to AL amyloidosis patients? What about that?
That I haven't seen any of the trials opening up for that, but yes, usually the amyloid patients, so that we know that for sure that daratumumab works very well for amyloid. So the cells that produce the light chains that then become the amyloid, it works. My assumption is that it would work very well for those. The other thing with amyloid is that these targets are not going to target the amyloid that's actually in the organs.
Yeah, they exist.
So exactly. So we'll target kind of stop the production and target the source cells, but it won't target the organs that are like the amyloid protein that is already there. So there are other trials with kids, for instance, KL101, which is another antibody that specifically targets the existing amyloid to kind of break that up and to reverse the amyloidosis in the organs. So, I mean, going forward, a combination of a bispecific antibody with something like KL101, that could be a really interesting combination. But I don't think those trials have started yet.
Yeah, because you wouldn't want to do just like an existing trial right now, just targeting the myeloma portion, and then because you're not affecting the
exactly. And we know that daratumumab and the CD38 antibodies, they're very, very effective in amyloidosis. So the major thing to get to in amyloidosis is really to get to something that will reverse the amyloid protein that is already deposited.
Okay, we have so many good questions. We have a lot of questions actually about smoldering myeloma patients. So how long, like if you foresee an approval in the next year, and I know for the CAR T, I don't think there's anything in CAR T for smoldering myeloma patients at this point, just newly diagnosed, maybe high-risk patients. How long do you think it'll take to consider this in the smoldering? And is it a good strategy compared to the other types of strategies for smoldering myeloma patients?
I think it's a really interesting question, and I think it would be a good strategy in terms of thinking about multiple of smoldering. I think it's certain strategies. One is, try to go as long as you can without any therapy, but then the goal is really then to kind of, with therapy in those trials, to try to prevent it from becoming asymptomatic and actually becoming symptomatic and becoming multiple myeloma. Still, that would mean therapy. So what you optimally want in smoldering is something that is not too toxic. But they're kind of two different strategies in smoldering as well. Some are aiming at giving something light and kind of just keep maintaining it at a level where it doesn't transform to myeloma. And some trials are aiming at kind of getting it, hitting it hard, doing triplet or even quadruplet chemotherapy, getting it down to 0, MRD negative, doing a transplant. So really, really kind of, we don't know which of these strategies is best. Both seem to be, obviously, in smoldering, treatment of some kind usually prolongs the progression to multiple myeloma, but that would also mean that you are on treatment. So, but I think these are really interesting for the smoldering space. I think that a lot of the smoldering trials are done with the monoclonal antibodies, isatuximab or daratumumab, because of the limited side effects that they have. The good effect with limited side effect. So that would be kind of the trade-off, how tolerable the side effects are and what each person is willing to do in terms of getting to a good response. If you either, it would be very much of a discussion with each patient. It would mean, yes, they have to be admitted for probably a number of days each week in the beginning and then come in weekly. That is still a large change in someone's life versus watch and wait and coming in to have labs every 3 months or so.
And I guess it's like the smoldering myeloma or high-risk smoldering myeloma, you're going to have two different strategies for that because you're active to progress, and you probably don't want to be in like a single line of therapy if you have high-risk smoldering and are going to go into active treatment. So I see what you're saying. Okay, Steve is asking a question, just a basic question, like what's the patient profile look like? I know you've got I love your slide. We're going to use that again in the article about sharing what you had to say. But what's the patient profile look like for most of these? Like how many prior lines of therapy? And you're not doing smoldering or MGUS or anything like that. It's relapse refractory myeloma. And is there an average like prior lines of therapy or?
So all of all of the protocols. So in the trial protocols, they're very strict inclusion and exclusion criteria.
Mhm.
So all of the protocols are written so that they should have 3 prior lines or more.
Okay.
And oftentimes it's also written that you should have seen or are refractory to a proteasome inhibitor, an IMiD, and a monoclonal antibody. So most of these patients, the average was that they've gone through 5 or 6 prior lines. So 5 or 6 types of treatment.
A lot of therapy, yeah.
That's a lot, yeah. And then also around 80% were refractory, not only treated, but with actually refractory to proteasome inhibitor, IMiD, and monoclonal antibody. So these are pretty fairly relapse refractory, kind of fairly far along patients that were included in these trials.
Right. And the existing drugs just aren't working for them. So that's what you're saying, refractory.
Okay. Yeah.
Okay, Paul has a question. If he's currently on BlenRep, does that disqualify him for these new bispecifics? Because same target, right? So we talked about that, and you talked about how BCMA can go away and then come back again if you have a space maybe of therapy. How would you answer that?
Most of the trials, I would say, allow prior BCMA targeted therapy.
That's nice.
Either belantamab or BlenRep, or you can go from a CAR T targeting BCMA and then to a bispecific. So yes, most of them allow for prior BCMA targeted therapy.
Okay, and do we have any inkling about how much these will cost? Laura is kind of asking that question.
I don't know. No.
Yeah, did you kind of compare it to, okay, and I'm on Revlimid or I'm on this triple combination, or that's hard to know.
I think unfortunately they're going to be pretty pricey, but I have no clue or no idea, or haven't heard anything of what's.
Yeah. Arjun was asking, interesting question, could these be used as maintenance therapy? So could you do, let's say you do a stem cell transplant, and you do, and it gets approved as a maintenance therapy. I mean, you'd have to get through a lot of trials for that, but I don't know.
Yeah, I think so for sure. I mean, we're doing now trials with, we have a maintenance trial where we're comparing Revlimid versus daratumumab. So I think that certainly certainly can be used at that point. Certainly also in some of them, and kind of what we're seeing a little bit with belantamab as well, is that, I mean, this is my own speculation, but could be that we have a more intense therapy, say every week, and then when we see a good response, maybe space out the therapy to a more maintenance strategy after a while where we see a good response. Certainly that has been done with, I mean, not as an official belantamab or BlenRep strategy, but in those that have ocular toxicities, we lower the dose and then we space it out instead of giving every 3 weeks to every 6 weeks, and that we still have a lot of patients that continue on a lower dose with more spread out scheduling. So I think that could be something that could be done for sure as a maintenance or as an intense part, and then moving to a maintenance scheduling.
Okay, and I have my question written down. I think I'm going to save it because we have so many questions still. Shirley is asking a question, and this talks about the T cell exhaustion. I want to ask, is there, because I think everyone wants to know this, like, how can I test and see if my T cells are exhausted? Because she's saying, if I tried a Abecma and I am relapsing on that, does this mean that these T cell engaging bispecific antibodies will not work for me? Like, can you tell them in advance?
No, no, there's nothing that's saying that you would not respond after Abecma, because Abecma is, what you're doing there is you're taking the T cells, you're reprogramming and then giving them back. So it's not those T cells that we are kind of recruiting, or it's not the CAR T that we are really, I mean, they might be, but that it's not specific that we're aiming to target those, or that those are the ones that become engaged with the bispecifics. So with the CAR T, what happens is that we see the CAR T in the blood for quite some time, and then they kind of disappear after a while. So we can measure the proliferation and how long they stay in the blood. But then the other it's really the other T cells that we're targeting with the bispecific.
Oh, okay.
So it's the non-CAR T, kind of the normal T cells.
Oh, so totally different set of cells. So it doesn't, you won't know. Is there any way?
No, you won't know that. Sorry?
Is there any way of predicting all of this? Can we take some kind of lab markers and say, you will do better on CAR T or bispecifics than somebody else? Or we just don't have that panel yet?
We don't have that panel yet, no.
Okay.
No.
I would like that as a patient.
Yeah, I would love to have that as well. I mean, going forward, at least what we're hopefully able to do is to look at more kind of the surface expression, and kind of maybe choose between, do you have more of BCMA or do you have more of GPRC5D or so in terms of that? But then bispecific versus CAR T, I don't know if there's a way of actually predicting if you would do better for either one of those. Yeah. I think it's so smart. If I've been through a BCMA targeted therapy, if there's a choice between BCMA and something else, it might be better to choose the other one and to kind of sequence them as well.
Yeah, interesting. Okay, Yvonne was asking about the success rate of patients with high risk deletion 17, but you don't have that data yet, right? Because the trials are so small. Yeah. Steve was asking kind of how you, like practically speaking, how you get into these trials. And I just want to make mention that, so we have a platform called the HealthTree Cure Hub. Patients can enter information into it. We have an integration with Spark Cures to help you find specific clinical trials. So instead of going, you can go on clinicaltrials.gov. You can search through those. It's going to be a long list, and they're hard to kind of navigate. So if you have a profile, then it'll match you with, it'll look at like your platelet count and your different creatinine level or whatever the inclusion criteria is for that trial. And then it'll show you kind of by geography and things like that, where the studies, these bispecific studies are open. So you can go to healthtree.org and create an account there and kind of go through that process. If you need help, we have a service that can help you, but you can always find them on clinicaltrials.gov as well. It's just not as easy.
Yeah, exactly. I think that's I just want to say, I think that's a wonderful, wonderful initiative. And we work with Spark Cures as well. So I think that kind of the platform that you and they have, it's really a wonderful tool. Yeah. And we are just starting to use that as well, because if there's something, I mean, we have, even though we're MSK and we have a number of trials open, sometimes if there's another center that's close by that has something that we don't have, we collaborate and kind of send patients to each other as well. So so we do the same thing within our own network from our end, and also able to look at kind of these platforms and these databases. Also, I think that's a wonderful, wonderful thing. And now I would also say with telehealth, we are very much open to second opinions and kind of discuss what we have as well. So we have a number of patients, that get all of their treatment. We have some that come for second opinions, and then we have an ongoing second opinion and they get treated locally many hours away. And then we have patients that come once and say, this is what we have. And then if they, live far away still, we can give advice and say that, or kind of say what we have to offer. So I think that's also kind of one thing, but to get a full overview, these databases are wonderful.
Yeah, well, in addition on the telehealth side, so I know during COVID, a lot of facilities opened it up and you didn't have these cross-border state restrictions or anything like that. Is that still in force for, like, if a patient in California wants to get a consult with you or something like that, can they do that, or are the laws?
They put some of their restrictions back, so it really depends on which state it is in. Most states are fine.
Ridiculous.
Yeah, I agree.
As a patient, I mean, I'm going to fly to come see you anyway, so what does it matter? It doesn't, I don't know. I think we need to stand up as patients and just say why, like for specific diseases, I could see how that's a problem, but in this disease, no, it doesn't make sense.
No, I fully agree. So most states I would say are fine. Some states we work it out so that we for that day get a temporary license and then it works out. But some states just say blankly no, and then yeah, we still find ways to get around it from our end. But yeah, it's state by state really. But I agree. I think I think it should be.
Yeah,
not restricted.
It doesn't make sense as a patient. Okay, Bonnie has a question. She said, you mentioned platform trials. I had written an article about Dana-Farber was starting to do, you talked about combination therapies, and this is her question. They had done a study with just a small number, kind of testing what to combine these bispecifics with, because most myeloma treatment is combination therapy, and we're using these things. And it showed that they were having the worst effects, like combining it with daratumumab, because it was doing something to the NK cell function or something like that. And then it was better with cyclophosphamide, which is kind of like this preconditioning therapy you do with CAR T cell therapy. So it seems kind of like ironic that you're using chemotherapy to make the immunotherapy more effective. But do you want to talk about that? Like when you're thinking about combination therapies, what's sticking out in your mind, and how do you, as an investigator, like conducting these trials, how do you go, well, this might be good logic, like around the combination therapies?
I think there are different ways of looking at that. First of all, there's certain things that like logically make sense, where you can have one thing kind of potentiate the other. So have this kind of, there's this one drug that kind of, as I said, that potentiates or can increase the expression of the BCMA. So I think that's a logical, interesting combination that makes sense. Other combinations that I think there's a lot of trial and error kind of going on in terms of all of these. But I think kind of combining in a smart way with kind of where it comes from, the mechanisms come from different angles, kind of. So you have mechanisms that complement each other, whichever that may be. So for instance, the upfront therapy where you have a 3-drug combination or 4-drug combination, all with different mechanisms, and you get deeper and deeper responses with kind of the more effective and kind of more drugs you add, at least for up to 4. I think it's a combination of both, because even though you can think out the smartest way of combining things, it might not even work when you
try
it. So a combination of smart logical thinking and then some trial and error, I think.
Yeah, okay, so we're just going to have to get practice with it. So we're planning on running a study, a real world data study, with participants who are in this HealthTree Cure Hub, to see, like, for CAR T and for bispecifics in the real world, looking at your prior lines of therapy, and then you'd use a bispecific or CAR T, like who's getting the best responses, and is that because of your genetics, or is that because of whatever? So everybody can kind of like keep that in mind, that we'll be opening that up sometime in the future.
Yeah, and it's really, really interesting.
Because that would help maybe you develop, say, oh, I don't have to run like 10 different clinical trials. I can see, well, maybe the data is pointing this way, and maybe we should open this study for combination therapies. Yeah, that's the goal, and that's the fun that we're having with having a platform like this, is that we as patients can contribute to you as researchers for a little more insight.
Yeah, no, I think that's wonderful. And I have a background in epidemiology, so I'm all for those. I think those trials, clinical trials that's the what we need to do, and that's where we kind of, everything is very controlled. But also it's a very select, because of these inclusion and exclusion criteria. There are very kind of strict limitations to the number of patients and the type of patients and performance status, and you said hemoglobin level, and kind of all the labs and everything. So the real world, where we actually see, the broader, we see everyone, those trials are very, very important, super important for, and you can include more patients, you can see more patterns, and kind of more of the details as well. So yes.
Okay, I'm not making a dent in, in the questions. I'm so sorry, everyone, but I'm trying my best. Okay, Linda is asking a question. Can you test for BCMA expression so you know if a second treatment targeting it could be useful? So you talked about, I know there's a CAR T therapy open, like at the Hutch in Seattle, that is adding this thing to kind of increase the BCMA expression. You talked about it already. Do we have to have increased, I mean, we'll test it and see, right? You'll get results back and you'll know if increased BCMA expression has an impact, but should people say, hey, what's my BCMA level? And does that make me a better or worse candidate for a bispecific BCMA targeted trial?
I mean, we for some of the trials, especially the CAR T, we have that, we test that. We test that. The ways of testing it are, we test with immunohistochemistry on the bone marrow plasma cells. So there are certainly also more sophisticated ways of testing it, and we're doing that as extra studies in the clinical trials, but we're not making decisions on that. But some of the trials, you need to have a certain level of expression to join the trial.
Oh, okay. I didn't know that.
For at least you have to be, you know, over a minimum threshold kind of thing. You have to have some there.
Okay. So you talked about, Sally has a question. You talked about the CAR T having kind of more physical criteria in terms of the study eligibility. On the bispecific, is there any age cutoff? Is there any physical limitation cutoff? Probably the standard, like you can't have non-secretory myeloma, I'm guessing, because I haven't looked at these study criteria. But what are the most limiting factors, I guess, in joining a trial like this?
So the most limiting factors, there's actually one of them, but I don't remember on the top of my head which one it is, that also includes non-secretory ones. So we're trying actively to include that, because we know that that's an unmet need as well. But most limiting, I would say, is if the myeloma is more advanced, then it's going to be hard to meet the criteria for the blood counts, renal function, that kind of thing. Also, you need to have a performance status of ECOG 2 or more, meaning that you can't be really all day bedbound. You have to have some kind of more activity. Otherwise, and that's more of a safety reason, because, as I said, these are still fairly tough treatment, so it might we don't want it to be too toxic for patients. Also, hemoglobin, for those counts, it needs, usually hemoglobin should be over 8, ANC, so neutrophils need to be over 1, platelets either over 50 or over 75, which one when it is. Creatinine clearance usually over 30. So if there is an advanced myeloma, those myeloma affects all of those counts, so that typically it's what's limiting to get into a trial.
Okay. There's a good question also that like some of the treatments, like maybe Revlimid or Melphalan that you're getting with transplant, can sometimes affect your DNA or cause secondary cancers. Is there any information or thought around any of the bispecifics having any impact like that? Or causing things like MDS or?
Not that we know. Also, we haven't used them very long. Most of the trials have been, the median duration is less than a year, and those things tend to develop over years after therapy. But in terms of the mechanism of action, since these target the surface and have T cells kind of have these granules and toxic molecules to kill the myeloma cells, it's a different action than, say, Revlimid and Melphalan, because their mechanism of action is to target the DNA.
Yeah, oh, right. And this one is to engage your immune system.
Exactly. So these that we know of, at least, do not target specifically the DNA. So they don't kind of induce that same type of mutations or so that we see with Melphalan or with Revlimid.
Okay, Chari's asking a question that was asked already as well. So if you're relapsing, you've had a prior transplant, you're considering, your doctor's recommending second salvage transplant, or you're looking at a CAR T or bispecific antibody, what would you suggest? You know, do you try the immunotherapy? Do you, because I know some of the, I don't know how it is on the bispecific antibodies. I know some of the well, maybe this isn't true. Like, is there a window you have to be post-transplant to join the CAR T or the bispecific? I think there is, right? Like,
I think there
no?
is. Usually it's at least 3 months, but
Okay.
it's usually
It's
not
that
very long.
Oh, okay.
I don't know if there, on the top of my head, if there's anyone that has more than 3 months. Might be more for the rare occasion that you've had in allogeneic, and that has been
Yeah. And well, you can't run
now.
trials if you do that anyway.
Yeah, well, most of them are actually starting to allow that now. So
Nice.
yeah. But I would say if a second transplant versus CAR T or bispecific, I would probably go with the CAR T or bispecific. Because it's a different action and something that the myeloma cells haven't seen yet.
Right. Okay. That makes sense. Somebody's asking, are there any bispecific trials going on in Europe?
That was a good question. I actually
I don't know the
where...
answer. Yeah, I don't know the answer to that either.
You can look on clinicaltrials.gov. You can see kind of all of the centers where it's open. I actually don't know if they've gotten to Europe or not yet. That's a very good question.
Okay, and as we think about, another question is about the immune system and its relationship to the microbiome. So I know we're working with Dr. Shah at your center to do some a study about precursor conditions and the effect of the microbiome, and people will be like contributing samples and things like that. Do you look at that? Like, is that being reviewed or tested, or should it be?
In a lot of the trials, it's being reviewed and tested, not so much on the clinical side, because we're still in the phase where we're kind of gathering all the data. And we know that there are certain bacteria that are associated with a favorable outcome and certain that are worse. And then the diversity is also associated with a better outcome or so.
Right.
We don't really know at this point if there's anything that we can actively do to get the gut microbiome to get to that point, kind of.
Oh, so like, what would you change?
Exactly, that's the.
You're not allowed to eat.
We know that eating lots of greens and fibers and so on, and especially the study with that Dr. Shah is doing, which is plant-based. I think that has seems to have a very good and good effect on the gut microbiome in terms of the diversity and certain specific bacteria. That seems to be a favorable outcome if you have. But we're still kind of it's a very, very interesting question. And everyone that I see, the first question, or kind of in the first, season one, what can I do? What can I eat? So there's a lot more to learn, and we're in all of not all, but in a lot of the clinical trials that we have, whether it's regarding diet or not, we certainly are including samples to look for the microbiome. So more to come on that for sure.
Yeah, that's a great idea. I like that. Okay, Robin's asking a question. How many patients in all these bispecific trials have had prior stem cell transplant? But if you're saying they've gotten like 5 to 6 prior lines of therapy, probably most of them, right?
Yeah, most of them. Sure.
Okay, so you're still seeing these high 65 to 89% response rates even post transplant.
Yep.
Okay, Susan's asking just a basic question, like, did any of the bispecific treatments include hair loss? And that is not
that I know of. No.
Okay. Vicky has a question about, and this is more a CAR T question than a bispecific question, since all the bispecifics are off the shelf. But she's asking about freezing T cells and harvesting T cells. Have you heard a lot about that, or is that a direction you think that people will head in, so that people who have had these earlier T cells, or when you say your immune system's not so exhausted or beaten up with multiple lines of therapy, can use things easier? Or do you think that's going to be a moot point because the therapy itself will maybe be available earlier?
I think that at least we are, there are some ideas of doing that at our center, at least in trials. When you collect the stem cells for the stem cell collection, actually collecting T cells at that point as well.
Mm-hmm.
So I think, I mean, in a number of years, I think that's what we're gonna do. Why go
that's
through
very
two types of I mean,
Two types
yeah,
of
I think, I mean, this is quite a
Yeah.
bit in the future, but yes,
Yeah,
I think that's what we're going to do. And there are initiatives of starting that in trial form right now as well.
Mhm. Okay, I like that idea. Okay, Glenn is asking, how does COVID relate to these bispecific antibody trials? Like if you're looking to join a trial, are there any considerations you need to think about during COVID? And yeah, okay, I have a follow-up question with that. It's an idea.
So not so much specifically. We tried to keep a number of these open last year. Also now everything is open, kind of as usual. I would say even more important to be careful, even more important to get the vaccine prior going into any of these trials. Also get the booster. We're recommending the booster now to anyone who is on active therapy or 6 months out from active therapy. Be careful. That's the main thing. These trials, as any of the myeloma therapies are, so both myeloma itself and the therapies, they do give a certain degree of immunosuppression, certain more than others, certainly more after a transplant and so on. But I would not hold off joining a trial or doing a therapy because of the threat of COVID.
Yeah,
Those that need treatment, go ahead with the treatment, because that's going to be more important than COVID.
And we're all very well versed about how to stay safe. I think myeloma patients out of transplant are some of the most well-educated patients
I know.
on how to wear your mask and do all that. And the only follow-up question I would have, and I think this will have to be the last question, because I have to give Audrey some time to make some closing announcements. But in CAR T, I know they're saying for CAR T patients, 3 months post, we're going to revaccinate you, maybe for everything, and including your COVID vaccine if you've gotten that. If you're on this continual bispecific antibody and you have lower, like, I don't know, did they even look at that in some of these trials, like COVID responses and patients on bispecific antibodies? Are patients on bispecific antibodies developing a COVID response to the vaccine? And then, like, when would you do that? Because how long do you stay on the bispecifics? Just it's forever, or no? Like, oh, okay, you just stay on it until it relapses.
Yeah. So I don't think that these are certainly going to affect the immune system. Maybe not as much as a transplant or CAR T.
Yeah.
So I would say we usually don't do the whole revaccination thing as we do after a transplant or so. So usually not that much, but we have certain ways of somewhat looking at at MSK. We have certain assays, and also we're just on a research trial of the immune responses after COVID vaccine. And we know that they're there, but compared to healthy controls, or non-myeloma controls that are not on treatment, do not have any type of malignancy, that the responses are not as strong. They're there, but not as strong. But these things are so difficult to measure, because we measure certain parts, but there are various types of antibodies in this trial that we've done. We haven't measured the T cell response, for instance. So there's a lot more to it. So I can't say anything different than we usually don't do the whole revaccination program, but get the booster shot as with all the other therapies.
Yeah, okay. And now approved for us as myeloma patients. This the third booster. So talk to your doctor about that. Okay, well, Dr. Hultcrantz, I'm so, you were just delightful and so knowledgeable. And thank you so much for answering so many questions. We knew this was not an hour-long presentation because patients have so many questions about these new therapies coming out. So Audrey, if you could copy all the questions, we'll try to get your questions answered offline, as many as we can. But I just want to thank you for joining us for this session today. It's fantastic, and we know a lot of patients will be interested in hearing from you.
Thank you. This was certainly my pleasure. Thank you so much for organizing this very, very important webinar.
Yeah, I just kudos to both of you for getting all those questions in. That was so fun to just hit them one after
Rapid
one.
fire.
Yeah, it was great. And thank you again, Dr. Hultcrantz, for sharing this time with us. Well, as Jenny mentioned, we're just going to finish with a couple outro announcements. So I'm really excited about our next immunotherapy treatment chapter. This will occur in October. We're still ironing out the details of exactly when that will be, but we will keep you updated. We're going to have a watch party to see the amazing documentary Jim Allison Breakthrough, followed by a Q&A session. So for those of you who don't know, Jim Allison is an American immunologist and Nobel laureate who holds the position of professor and chair of immunology and executive director of the immunotherapy platform at MD Anderson. His story is amazing, and his research has helped develop many immunotherapies we see being approved in the cancer field, including CAR T cell therapy. So we will keep in touch about when that is, and we are excited to have you join that interactive event. You may be interested in other Myeloma Crowd community events we have coming up. On the 1st of September at 7 p.m. Eastern, we'll be discussing the Myeloma Move Spring event, which is a 30-day fitness challenge, and talk about how we can stay motivated throughout the month as we honor Blood Cancer Awareness Month in September. On the 2nd, we have 2 events. Our first is a stem cell transplant event at 1 p.m. Eastern. Multiple myeloma journey partner Michael Riota will share his experience about what happens when stem cell transplants don't go as expected. Michael experienced an infection, some other things as well, but his medical team really took care of him, and he wants to share his experience because, though rare, we want to give a realistic expectation for people about what the stem cell transplant might be like. At 7 p.m. Eastern that night, we will hear from Dr. Jens Hillengas about why he has decided to study the relationship between fitness and myeloma progression in our MGUS Smoldering Myeloma chapter event. On the 7th at 1 p.m. Eastern is our Myeloma Financial Coach chapter. Diana Valentine's talking to us about good debt, bad debt, and how that affects us as myeloma patients and caregivers. At 7 p.m. Eastern on the 9th is our Myeloma Caregiver chapter. Licensed social worker Nora Miller will be discussing how we can make boundaries and keep them for the benefit of everyone. And on the 14th is our SoCal Myeloma Community chapter that starts at 6:25 p.m. Pacific and will feature myeloma specialist Dr. Fotis Asimakapa.
And we just call him Dr. A because he's awesome. And his name is difficult to say.
Yes. The link to sign up for any of those events and even more events that I haven't mentioned, which I know I mentioned a lot, is found at the bottom of the slide and will be sent out in that resource email you will receive along with the recording within 48 hours of this event's conclusion. Again, a thank you to our sponsors, Bristol Myers Squibb, Takeda Oncology, Karyopharm Therapeutics, Adaptive Biotechnologies, Oncopeptides, Janssen Oncology, AbbVie, Sanofi, Amgen Oncology, and Genentech. And thank you to each of you for joining with us today, helping us build a stronger myeloma crowd community. We appreciate each of you and hope that you have a great rest of your day. Thank you.
Thanks to all of you. And thank you, Dr. Hultcrantz, again for participating. We appreciate you so much.
Yeah, thank you.
Thank you, everyone.
Bye-bye.